All right, so in our third and part of this quantitative genetics lectures, I just want to go over some measurement techniques that have been used in humans and effectively in humans, okay? If you're doing agricultural things, there's other ways to do it, and there's certainly other ways to do it in humans with lot scores and other things. But I just want to go through some of the classic ways that it's been done, okay? Well, really what we're trying to do, again, is trying to go after this narrow sense heritability, which is this additive variation divided by the phenotypic variation. And one of the ways we can do this is by looking at this offspring relationship to parents. Okay, so yeah, we can do this by in many ways, but basically taking the average of the parents or some information about the parents with the height or the intelligence or whatever blood pressure rate or whatever, and comparing that to the progeny that they have and what the progeny have. And this is where pedigrees come in because, handily sometimes, because you can get information about the grandparents and the parents, and you can make a much bigger and broader scale of this. So actually, they do use pedigrees for this quite frequently. Okay, so what's the relationship here? Well, let's take about mid-parent height, okay, the average height of mother and father. So you want to use this because if dad's 6 foot and mom's, 5'8", which one of those, well, they both contributed, so it's going to be somewhere. So we tend to take the mid-parent, the average of the two, and we plot it, okay? If you have heritability that's, flat like this, the line that fits through, best line that fits through the relationship between the progeny and the parents is flat, you really don't have any broad sense heritability. If you get a line that's got some slope to it, it looks something like this, but it's got variation around it. Again, the variation is along this line, right, and off of the line. About 50%, about what you expect. We saw that in some of the levels of traits we talked about. The narrow sense heritability around 4 to 6%. Or 60%. And then heritability, if you're really tight, if you say, I can tell you based on the mid-parent height exactly what the child's going to be right here, okay, and predict that strongly, then I've got a heritability that's quite high. Okay, so how do you really get to that? Well, mathematically, you get to it by the covariance. Those of you who had any statistics have run across covariation. The covariance between any two variables, okay, it's this little equation. You just take each x of I is just each little point on x, subtract the mean for all the points on x. Y is the same way. You find a y point, you calculate the mean for all the y variation, and you sum all that up, divide by the n. That gives you the covariation between them. Things that are flatlined have no real relationship will have a covariance of 0. If they point in a positive direction, in other words, as things get taller here, the progeny get taller, as the mid-parents get taller, you'll have a covariance that's positive in some way. Or if the children get shorter while the parents get taller, then you'll have a negative covariance. Okay? Now, this is useful because you can figure out the slope. Now, monozygotic twins are hypothesized, at least, on a purely additive fashion to have almost a one-to-one relationship, right? Okay, you don't want to worry about the negatives down here. But monozygotic twins, full sibs, okay, your brother or your sister, somewhat less than that because you don't share all that same information. Remember, you've got recombination going on, so you didn't get the same genes as your sister did. Parent and offspring is a line of about 50%, and then half sibs, someone that's made like this. And so the slope, what slope determines this is the covariance divided by the variance of x, okay? Whatever variation in the mid-parent divided into the covariation between the mid-parent and the progeny offspring, okay? So where's this useful? Well, we can, by looking at this, we can define some of these components. The additive genetic component, which is narrow sense, right? Can be used to make narrow sense. The dominant genetic component, and then this environmental component. Monozygotic twins, the covariance between them and their parents is a combination of all those, and so you can parcel those out by some sophisticated techniques. Parent to offspring gives you a direct measure of the variance that's additive. That's why it's so often used. Okay, it's 1/2 the variation that's additive is parent to offspring, 1/2 because your genetic contribution is 1/2. Full sibs have a more complicated model, but you can work with it. And then half sibs have another model. So of these, the monozygotic twin models are used frequently because we're interested in all of these. We want to pull out all those components. But if we want to just get to the heritability part, the narrow sense heritability, parent offspring is very effective. Okay, so here's a little example. I would put a big star, red star, by this particular figure table because it tends to show up on things like midterms and stuff. Okay, I don't memorize it by any means, but look at it and know quickly how to work through it. And lots of problems and problem sets. If you're told about an offspring parent and I want to know the heritability of some trait and I have that information, I can give you the covariance, or I can give you the slope of a line, and then or make you plot a line and tell me what the slope is or something. And you don't have to do like calculate like that. I mean, you can do it on a computer. You can get the slope just quickly from anything. But basically, the covariance gives you these measures of broad sense. Okay, and then if you want to directly go to the narrow sense heritability, for some of these, you could just multiply 2 times the slope or 4 times the slope, or if you know the mid-parent, all you need to know is the slope. Okay, heritability is the slope. So once I calculate the slope, I can calculate the heritability. Right, and that's really one of the ways it's done. Is that we look at cholesterol levels, we look at hand size or whatever traits you might be interested in. Figure out what the parents have, mid-parent, figure out what the progeny has and offspring has, and then you can work from it, provided you get enough numbers. And you saw that one of the studies that we brought up, broad sense heritability or narrow sense heritability, 2,000, 3,000 cases that were looked at. That's when you start getting reasonable estimates. Okay. All right. So the last thing I wanted to tell you though is that if you start thinking about heritabilities, different traits, remember when we went back and we talked about the types of traits that when we talk about history of genetics, types of traits that you often look at. Some are morphological, classic phenotypic traits. And we said, okay, those are good, but then you can do physiological traits, where you can go in and modify them and look at them. Okay. Then there's things like behavior, which we're going to cover in later lectures. When you what's the behavior function, and then things like life history. How many progeny can you produce? What's the success rate of those progeny, etc. Those are life history traits. And the heritability estimates of those, the morphology, just like you would expect, the cumulative amount of heritability of that is less because there's a huge environmental input on it, okay? And you can get it. Physiological also has an environmental input, but we're programmed in our genetic sense, and it's very close to what behavior is. Life history peaks out pretty quick. Okay, notice life history peaks out at about 0.4, 0.5, and then it doesn't do much, accumulate much after that, meaning that there's not a lot of life history variation out here at 1. Okay, there's a lot of environment. It quickly goes whatever genetic components there are, and then everything else is environment. For morphology, you still get a continuous change depending on the traits that you're looking at. So these are ways that it's been used to look at differences in human groups, different relationships to humans, to altitude, to all sorts of food and those sorts of things. All right, well, that basically finishes up everything that I wanted to say right now about quantitative genetics. Again, I was trying to give you some terminology, some basic ideas about what traits might be looked at and what kind of levels of heritability you might see. We'll come back to these again as we work through some more of the lectures in the second part.